Hi, I'm Dr. Ohnishi from Hokkaido University, Japan. Today, I'd like to present our research into the effect of gender differences on peripheral ghrelin secretion in aged mice. As you know, ghrelin was first isolated in 1999 from the rat stomach as a peptide hormone with a powerful appetite-stimulating effect. The concentration of peripheral ghrelin is known to show different dynamics in males and females. However, variations in ghrelin levels in aged males and females have not been studied. In this study, we investigated the differences in peripheral ghrelin secretion in aged male and female mice and analyzed the underlying mechanisms. We used C57 black 6 mice aged 80 weeks and 6 weeks. We first measured food intake after 24 hours of fasting. As you can see in this figure, food intake in males was lower in the aged mice than in the young mice, whereas in females, there was no difference between aged and young mice. Therefore, a decrease in food intake with aging was seen in male mice only. We then measured ghrelin concentrations in blood samples taken during fasting and feeding by ELISA. Here you can see a typical ghrelin level change in young mice, namely an increase in fasting, decrease in feeding state. In aged male mice, however, ghrelin level in fasting decreased significantly in comparison with young mice, and fasting-associated increase was not observed. Contrary to this, aged female mice showed high plasma ghrelin levels in both fasting and feeding, in comparison with young females. We next examined the expression of appetite-related genes and ghrelin-related genes in stomach and hypothalamus. The expression of the peripheral ghrelin gene in stomach was significantly increased in both males and females of the aged mice, and the expression of the gastric ghrelin acid-modifying enzyme gene also showed an increasing trend. These results suggest that ghrelin synthesis did not change with advancing age as well as gender difference. On the other hand, in hypothalamus, the expression of orexigenic genes such as NPY and AGRP were significantly decreased in aged male mice, but showed an increasing trend in aged female mice. Moreover, aged female mice showed a significant decrease in anorexigenic POMC gene expression. From these results, the appetite-related signals appear to be anorexic in aged male mice, but not in aged female mice. We then investigated the effect of Rikunshitou, a Japanese traditional medicine, which acts as a ghrelin enhancer, on the anorexia of aged male mice. Aged male mice receiving Rikunshitou showed a significant improvement in their decreased food intake. In summary, aged male mice showed a decrease in food intake. In contrast, food intake of female mice maintained with aging. This is probably related to the sustained ghrelin secretion, which is independent of fasting or feeding. Furthermore, we also found that Rikunshitou, a Japanese traditional medicine, improves the decrease in food intake in aged male mice. Our results may provide new insights into the mechanism of gender difference in anorexia accompanied with aging, and Rikunshitou may help improving appetite of those aged males. Thank you.